I'd like to explain the knock-in animals are useful in place of the cynomolgus monkey in GLP tox study. So agency has accepted our concept. So today's agenda. First, current situation and future analysis. And secondly, we propose a solution. And finally, I will mention the merit of the knock-in after KI animals. Please note, today's talk is my private opinion based on the information from our all parties concerned. So before my presentation, I'd like to introduce myself. I worked for Takeda pharmaceutical company for more than 30 years, and I have been global drug safety certificate lead up to 2017. So I have more than 30 years experience for toxicology. As a social activity, I have both board certifications in toxicology of the US and Japan. And I was also the president of the 48th Japanese Society of Toxicology annual meetings in 2021 in Kobe, Japan. So during the Takeda period, I was able to launch Takeyama in 2015 as a global drug safety therapeutic area lead. So Takeyama is a first-in-class medicine. Takeyama is a potassium competitive acid blocker. And also the Takeyama is fourth place in 2022. So gross sales is about $1 billion. And Takeyama is available in 6 Asian countries. So let's get started. First section, our current situation and the future analysis. So just as shows approved nucleic acid drug classification in April 2023. So as you know, nucleic acid drugs are classified into 4 types. Aptamer, messenger RNA, siRNA, and antisense oligonucleotide. So cynomolgus monkeys are utilized in almost all repeat tox studies of aptamers, siRNA, or antisense oligonucleotide, except for messenger RNA vaccine. So why monkey is used in the first place? So because agency requires to evaluate the on-target toxicity in non-clinical tox studies. So as you may know, in tox studies of new modality, that show less likely to produce off-target tox. So therefore, animal which show efficacy should be selected. And also, monkeys are highly homologous, intersecting with humans. So why high price of monkeys and why restriction on the number used? Because we considered 2 reasons. First is export suspension from China. Secondly, export restriction to Japan from Vietnam and Cambodia. So why is increased price in cyno? So because growing demand for far beyond supply. This is as you know, the impact of the new modality medicine. So maybe they concern about current situation. So it is undecided when and how many monkeys are allocated to research field. And there are no prospect of when this situation will improve. So this situation in Japan is very similar to the United States. So is this situation similar to South Korea? Or is this situation similar to your company? I think some researchers in Japan mentioned that small number of monkey can be secured, but the cost of animal cannot be paid because increased prices. And the development costs are very limited, and in a high priority project, monkey can be used. However, low priority project suspend or terminate. And your project also deprioritize. So another researcher mentioned that the good targets was selected or compound selected, selection is completed, but they cannot secure monkeys and they cannot prepare commission cost. So no future, it's all over now. So development of following new modality drugs may be suspended if the monkey cannot be used in GLP tox studies. For example, antibody and antibody drug conjugate and nucleic drug, siRNA, antisense oligonucleotide, and peptide conjugate oligomer or heteroduplex oligonucleotide. So here is the cost of development up to phase 1. So if please look at the right side, cost and number of animals to phase 1 is calculated. So if you use 10 monkeys for pharmacology study, 4 monkeys for DMPK, 10 monkeys for 2 weeks non-GLP tox, and 26 monkeys for 4-week GLP tox. Total is 50. So if one monkey cost $35,000, you need to prepare $1.7 million for monkeys, only monkeys. So if one monkey costs $70,000, you need to prepare $3.5 million for monkeys. So in addition to this cost, you need to prepare the study consignment fee for rats, monkey, genotox, and safety pharma, plus bulk synthesis fee, including a GLP bulk and analysis fee. So I'm afraid that academia and venture research activity would stop. So next, we discuss solution. So I propose 5 solutions today. First, you prepare the sufficient research funding. So big pharma has a lot of funds. So they continue to use monkeys. This is one. First. Secondly, GLP studies in China. There are many monkeys in China. So you maybe conduct tox studies in China. Number 3, use marmoset as a substitute for cynomolgus monkeys. 4th, wild type rodents plus dog. And number 5, use KI animals. So combination of the KI mice or KI rat plus dog. So utilize the KI animals that express human receptor. So KI mouse and KI rats exhibit pharmacological effects. So is it possible to solve the problem of high price and supply shortage of cynomolgus monkey? So next is I explain merit of the KI animals. So here is a comparison between the cynomolgus monkeys and KI rodents. So first line, look at the first line. As for the homology, so high in monkeys and 100 identical in KI. And next is on-target and off-target toxicity is detected in monkeys and KI. And NOAEL and tox dose for phase 1 are available in cynomolgus monkeys and KI rodents. And the phase 1 initial dose will be calculated from NOAEL in cynomolgus monkey and KI. And the metabolic tox and impurity tox are also available in both species. So required amount of the GLP bulk. So as for the cynomolgus monkey 46, 460 gram, and KI is 40 gram. So the required amount of GLP bulk is 10 times higher. And as for general tox and safety pharma, so monkey have enough background data, but the KI have a limited background data. This is the demerit to use a KI. However, reprotox, juvenile tox, and carcinogenicity studies, if agency requires these type of toxicity studies, maybe KI is possible to conduct these studies. However, in monkey, so reprotox of monkey is possible, but not feasible. Juvenile tox is also the same. And carcinogenicity studies is not possible in cynomolgus monkey. Next, this slide shows a comparison between the wild type rodents and KI rodents. So first line, on-target tox. So in wild type rodents, on-target tox is not detected, but KI, on-target tox is detected. Off-target tox is detected in wild type and KI, and NOAEL and tox dose is available only in KI. And especially, the calculation for the initial dose in phase 1, Mabel approach will be used in a wild type, and NOAEL approach will be used in KI. So, as you understand, Mabel is the minimal anticipated biological effects level, and NOAEL is non-observable adverse effect level. So, in case of a Mabel approach, how many cohorts should be planned in phase 1? Next, this is a big impact on the development cost and duration. I explain the next slide. So this is a phase 1 cost and duration comparison of the Mabel approach and novel approach. So we calculated using a 9 cohort of Maybell approach and 3 cohort of novel approach. So look at the general medicine. So in price of the general medicine, $1.7 million to $2.7 million will be needed in Maybell approach, but 0.7 million to $1.0 million in novel approach. To look at the duration of the general medicine. So it takes 9 to 15 weeks in Maybell, but 3 to 5 weeks in novel. So in price of the duration of the chemotherapy, similar trend will be shown. So if you choose a Maybell, phase 1 cost is 3 times higher and duration is 3 months longer. Next, many pharmaceutical companies concerned that the agency accepts inAnimals. So answer is shown in ICH S6 guidance. Look at the ICH S6 guidance. So animal models that are thought to be similar to the human disease, these animal models including transgenic animals. Transgenic animals means the inAnimals. And these models may also be useful in the determination of the safety. And next, this is a copy of a daily newspaper on September 11th this year. So PMDA senior specialist in toxicology, Dr. Maki, explained inAnimals as an alternative to monkey. And PMDA, Dr. Maki has accepted inAnimals for the GLP tox studies. Next, this slide shows our first American College of Toxicology and Japanese Society of Toxicology joint session in Kobe, Japan, and virtual in the United States in 2021. So I was a chairman and organizer, and I discussed recent topics with FDA and PMDA. Dr. Maki is here, and so Axcelead has continued to collect the latest information from the FDA and PMDA. So this slide shows CRO achievement in 2019 to 2021. So big pharma project may already be used to KI mice to advance to phase 2. So I explained. So first 3 lines, KI mice, 2 weeks tox, non-GLP tox was conducted. And 4 week tox, GLP 4 weeks tox, and 4 weeks tox background data. These 3 studies means this compound to go to the phase 1. Next, 13 week GLP tox, 13 weeks tox background data, and non-GLP embryo fetal development range finding, and GLP embryo fetal development, and embryo fetal development background data they conducted. This means that this compound moved to the phase 2, and then they are conducting 26 week tox background data, and 2 years carcinogenicity studies background data. So this means this compound candidate is very successful. So from this information, agency is likely to admit KI mice. So here is a timeline with the KI mice and rats. And first, so we recommend the producing a hetero animals for 7 months and conducted preliminary pharmacology studies in hetero. So if hetero animals shows efficacy, we move to produce the homo animals and then conducted pharmacology studies in homo. So during the pharmacology study, so we produce homo animals for tox studies. So first, single TK and 2 weeks repeat tox will be planned, and then conducted 4 weeks GLP tox. So remember, there is no impact on the timeline of IND enabling studies in KI animals. We can conduct it 9 months for IND enabling studies. So rate limiting is the first 7 months for preparing the knock-in. So quick start is highly recommended. So next is the IND package cost for general medicine. So if you selected knock-in rodents plus dog for repeat tox and genotox and safety pharma, total IND cost is $0.9 to $1.2 million. However, if you selected the cynomolgus monkey, maybe wild type rat plus cynomolgus monkey for repeat tox and genotox and safety pharma. So total cost is $2.4 to $2.7 million. So if you choose the KI, maybe you can save approximately $1.5 million. And you can do one more project. So here's the conclusion. So KI mice and rats can solve issue of high price and supply shortage of cynomolgus monkeys. So we encourage you to consider using a KI animals before suspending or terminating a project. So next, I explain smart IND briefly. So what is smart IND? So S means a specification, proposal of a concrete plan and method and goal. And M means minimum. Application of the minimal necessary requirement for an IND. A means acceleration, shortened development time. So R means a reduction, reduced R&D cost. The T means a timely proposal of strategy in line with our latest trend. Next, so fast is fast, smart IND. So what is fast is fast. So usually we conducted GLP 4-week stock studies just for 6 months. I a little bit explained about the timeline. So first 1 month, study starts after study director signs the protocol, and then pre-treatment period starts. And the next 1 month is the dosing period for whole week. And then after the completion of the dosing period, necropsy is performed. And after necropsy, data review, except for histopathology and TK, is done within 3 days. And 2 months after necropsy, histopath is fixed. So draft final report will be produced within 3 days after histo data fixed. So 6 months later, IND is ready. So what is first? The first is to summarize the final report. So board certified toxicologist and pathologist would add the mechanism of tox change in the final report. That's my presentation. Thank you very much. Thank you for hearing. I will listen to my presentation.
I'm Michiyasu Takeyama. I lead the genetically modified animals in Axcelead Drug Discovery Partners. Today's topic is knock-in mice or rat in Axcelead, key feature and uniqueness. This slide shows the purpose of using genetically modified animals in drug discovery research. Genetically modified animal is most traditional is knockout mice or rat. This knockout mice or rat is used for analysis of target gene or validation as a drug target. And action of the compound is on target or not, or immunize the animal for production anti-human protein antibodies. And knock-in mice or rat. These knock-in animal used for mimic of human genetic mutations or in vivo evaluation of small molecule with specific differences as species differences, or in vivo evaluation including nucleic acid or genomic sequence and mediated knock-in. This slide shows the method for creation knock-in mice. Knock-in mice can be categorized into relatively small DNA, human cDNA knock-in, or relatively large DNA, human genomic DNA knock-in. Human cDNA is a relatively small, maybe 2 or 3 kilobase. It's easy to generate the knock-in mice or rat for combined with genomic editing, CRISPR-Cas9. And also, if you use these knock-in animals for evaluation of the ASO or shRNA, in this case, occasionally, ASO target is into intron or non-coding area on the gene. So they have to including genomic structure, exon and intron for the human gene. We can create such a type of knock-in mice with under 40 kilobase using ES cells in mice. These human DNA knock-in mice rats are used for evaluation of drug with species differences. This paper described a case in which human GnRH receptor knock-in mice. It's generated and used for in vivo pharmacological study. This GnRH antagonist is a species differences in their actions. So the pharmacological effect on human GnRH receptor antagonist couldn't be evaluated in wild type mice. So we make the human GnRH receptor knock-in mice were generated by a knock-in of human cDNA into the mouse GnRH receptor gene. It's a replace of the human mouse gene with human gene. And these type of this knock-in mice can possible to evaluate the pharmacological effect in antagonist here or here. Next, this paper described a case of the human mutation knock-in in type 4 collagen gene. This mutation caused the Alport syndrome in human chronic kidney disease. So we knock in the mice type 4 collagen gene in a nonsense mutation. Its mutation is the same as in human. Generation of knock-in mice is the same nonsense mutation in exon 21 of type 4 collagen gene. It is a human patient with Alport syndrome. It's a familiar chronic disease. So this mutation causes the loss of type 4 collagen protein from glomeruli and hyperalbuminuria and short lifespan compared to wild type. It's a similar changes in human disease were observed and confirming the appropriate animal model. We collaborated with the Professor Noz in Kobe University. They have exon skipping technology using antisense oligo. They use exon skipping oligo. In this case, exon 21, there is a nonsense mutation. It causes the short protein from this mutation gene. However, if they skip the exon 21, it causes the short deletion of the whole protein, and most of protein can be recovered from this exon skipping. This antisense oligonucleotide administration, hyperalbuminuria improved dramatically, and lifetime is improved, and it confirmed that this the exon skipping effect. And this means the pharmacological effects were confirmed in a knock-in mouse and this animal model. We have also have the reproduction engineering technology, IVF, in vitro fertilization technique. It uses in short time, within F0, it generated the first generation of the knock-in mice. But about 9 to 12 months, we can propagate the number of the mice using this IVF technique. And we also have the lineage preservation, so the test can be a smooth resume after a certain period of the interruption. And in Axcelead, there is a many combined the solutions in one stop. So we can design the best genetically engineered mice and design for your purpose. And we propagate the large number of the mice for use the test in short time. After that, we can get the pharmacological test or safety test or biomarker discovery or such studies. Because Axcelead has a CRISPR license, ERS and both licenses we have. So you can feel confident in requesting a study using these genetically modified animals using CRISPR-Cas9. And also, our facility is located in the Shonan IPAC. It's a ALAC international certified facility. Thank you very much.